This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 622, recorded on June 1st, 2020. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Austin, Texas, Rich Condit.
Hello there, Vincent. Just briefly, 80 degrees, partly cloudy, looking like for rain in the afternoon. It is, rain's good. We got an excess of rain this season, and this gives me an excuse to stay inside and read and not worry about the garden, so that's good.
Also joining us from Madison, New Jersey, Brienne Barker.
Hey, it's great to be here. I'm a little envious of Rich's warmth. It's 65 degrees here, which is 18 C, and just generally seems kind of chilly.
Our guest today is from Hamilton, Montana, Rocky Mountain Laboratories of the NIH, where she is Chief of Molecular Pathogenesis in the Laboratory of Virology. Emmie de Wit, welcome to TWiV.
Thank you very much. I just frantically checked my phone. It's 15 Celsius here.
It's chilly.
I haven't been outside for a while.
You're up in the mountains, right? Well, you're in a valley,
Yes.
actually, right? Yeah. I was out there a couple of times to visit your lab. Marshall Bloom invited me a few times.
Mm-hmm. Yeah.
And I remember looking out his window, which you can see the mountains. Can you see the mountains out of your window?
I am just a few door downs from Marshall, so yes, definitely.
Okay. It's funny, I never saw you. And I guess I was there twice or three times. I never saw you.
I think last time you came out, I wasn't here. I was traveling.
Okay.
So.
And then, of course, I saw you in Singapore in December.
Yes.
At the NEPA meeting, where nobody was thinking about SARS, right? But it was circulating already.
Yeah. Well, it's funny. Around that time, I actually gave a talk in Bozeman at the university there, and someone asked me what the next big outbreak was going to be, and I said coronavirus. So that was sometime in November.
Wow.
So,
Nice job.
yeah, I guess it's all my fault.
Yeah, I was going to say, watch out, people will blame you.
Yeah, I wouldn't take responsibility for this. That could be dangerous.
So we're going to talk about your work, but before that, let's hear your history. You're obviously from the Netherlands, right? That's where you were born.
Yes, I was born in the south of the Netherlands, and then I did my PhD in Rotterdam at Erasmus Medical Center with Ron Fouché.
Who's been on TWiV a couple times, yeah.
And you
He got
studied.
pretty
You
famous.
studied.
Yeah, he did.
You studied influenza for your PhD.
Yeah, I did. I actually, when I started working with Ron, he had just returned from Philadelphia, so he had just switched to influenza himself. So it was actually quite exciting to kind of pick some topics to work on. And we decided that I would work on the packaging of the gene segments of influenza, which at the time, a lot of people thought that so influenza has 8 different gene segments, that they were packaged randomly when the virus was budding, and it would just package a few extra, and that would make sure that most of the virus particles had 8 different gene segments. And we were convinced that that wasn't the case. So that's how I started working on influenza.
And you were right?
Yes, correct. Yes.
Ron was last on TWiV in Rotterdam. I was there, what was it, there was a Congress of Virology meeting that Marion ran.
Last year.
Was that last year? Boy.
Mm-hmm.
Yeah.
I was there too, yeah.
You were there? Okay. Yeah. And so were you always interested in science growing up, or is that something that came later?
I know. I think I was always curious, but I never really thought I would grow up to, I never put much thinking ahead. Still not. I just go with the flow. So I was in high school. Yeah. I was in high school, and I really liked biology. So I decided to study biology. And then I think what I really liked was just the fact that it explains so many things that go on around you, right? If you're interested in behavior or plants or viruses, biology kind of covers it all. And then, when I was in college, it was the thing to do. After you graduated, you would go into grad school. So that's what I did.
And so after your PhD, what did you do next?
So after my PhD, I stayed with Ron for another couple of years.
Mm-hmm.
My mother-in-law was very sick at the time, so we decided to stay in the Netherlands for a couple more years. So during my PhD, even though I was originally working on packaging, there was an outbreak of highly pathogenic avian influenza in the Netherlands, which was pretty big. Actually, at the time, there hadn't been that many big outbreaks, and this was 89 human cases. So I completely switched the topic of my PhD to study the virus. There was only, out of those 89 cases, there was only one fatal case. So I started studying the pathogenesis of that virus. And so that's actually the main part of my thesis, I would say. And so that kind of got me really interested in these emerging viruses. So after I graduated, we decided that the next big thing in flu was transmission and how avian flu viruses become transmissible. So the transmissible H5N1 story actually started with my postdoc, but I left before the virus was transmissible.
So that outbreak that you described, I'm assuming there was not actually any human-to-human transmission in that. Is that correct? And if so, how did 89 cases arise?
So there was some suspected human-to-human transmission, so most of the cases actually had conjunctivitis. And so there was a family where they were using the same towel, and then there was a mother and a daughter that were using the same eye makeup. So they think that there may have been some transmission there. But this was just a huge outbreak in poultry, and a lot of the poultry workers, and then some of the veterinarians and the people that came in to get rid of the chickens got infected.
Was this a lethal infection in the poultry?
Yes. Yep.
Okay.
What's the subtype of that virus?
H7N7.
Okay. I remember that because there was some idea that it was infecting cells in the eye, which have alpha-2,3 sialic acids, right?
Yeah, that was one of the things that we really wanted to study, because that seems to be something that H7 viruses do more than others. But we never got around to finishing that.
And so you left before the big controversy over the H5N1 ferret studies, right?
Yes, I did. Yeah. So when I was working on that project, so what we did was we started by taking the things that we knew by then. So we knew by then that the virus had to make this receptor switch, and that it was really important to get as much virus in the upper respiratory tract as possible. And so we were making mutations in the HA to make sure, and then checking like what the where in the respiratory tract the virus would be binding and where it would replicate. And then we were also making mutations in the polymerase that would make it replicate more efficiently in mammals or humans. So we were doing all of these steps where we knew what had to happen. And all this time Ron was saying, just passage it in ferrets. And I was saying, no, I want to do this. First, I want to explore all of the options that we actually understand what is going on. So by the time I left, we had made a virus that was binding in the upper respiratory tract, but it wasn't transmissible yet.
We spent a lot of time on TWiV defending Ron and also Kawaoka, their experiments, because as you know, there were many people who started attacking them, who are around to this day and still are vocal, which is crazy. But on TWiV, we defend them because science needs to progress.
Yeah, I think one thing that got lost a little bit in the discussion is that this was done with a real scientific interest, because the consensus at the time for a lot of people that were very vocal was that only H1, H2, and H3 could transmit between humans, and that there was just the risk of H5 to become transmissible was negligible. And so that was a real question that we were trying to address. And I think that got lost a little bit in the discussion over the safety of these experiments.
There was a lot of the science that got lost in the discussion, you know.
Yeah, for sure. And because of that, there was this moratorium in the U.S., right?
Yep.
And it included some coronavirus work, right? some.
I think it was for a while. It was any gain of function studies.
Yeah.
And I know that Ralph Barrick said, you can have your moratorium, but nature doesn't care about it. It's going to keep progressing, which is true. So you left the Netherlands. You went directly to Montana. Is that right?
Yeah. I came here to RML to work with Heinz Feldman. So I was in his lab for almost 10 years, first as a postdoc and then as a staff scientist. So I started here on some BSL-4 viruses. So I worked a little bit with some Ebola viruses, mainly Rhesus Ebola virus, but then I got really interested in Nipah virus because it has that respiratory component that I knew from my time in flu, but then it also has this neurological component, which was really new to me. And yeah, I think it's one of the most interesting viruses. If you are like me and you like viruses that cause really severe disease, and if you like viruses that cause respiratory disease, then Nipah is quite interesting.
That's right. So I've looked at the list of viruses you worked on: Nipah, Ebola, SARS, MERS, high path influenza. So do you like? And you just said you like dangerous viruses, right?
Yeah, I think it's just the fascination with how does that work, and especially the negative strand viruses, right? Their genomes are so small. So how is that possible? What needs to happen for someone to get that sick?
Yeah. Did you, I think I saw in some papers, you were on some papers on the VSV Ebola vaccine. Were you involved in that?
I was really not involved in that. I have done I guess I did a little bit of work on the sidelines. So one thing that I did early on, so when I moved here, they had just found Ebola arrestin virus in pigs in the Philippines. So that was going to be what I was going to focus on first. So I had done, so as a sort of we wanted to do, to infect pigs with Ebola arrestin virus to see if they would actually get sick. But as a practice round, of course, we don't like to work with these known models in BSL-4 without figuring out what the potential problems are. So as a practice run, we tested if the VSV Ebola vaccine actually caused disease in pigs, because there's always a risk of the vaccine virus potentially spilling back into other animals. And then, of course, with a VSV, you could have a huge problem because it's a notifiable disease. So we didn't see any of that. But then it took so long to get permission to work with select agents in pigs that I kind of decided to work on Nipah instead. So that's kind of the extent of what I did. What I did there. We are also working, or at least Heinz is, and I did a little bit of work on that too, on a Nipah vaccine that is based on the VSV Ebola.
So when you went to Hamilton, were you any concerned at all that it was such a small town?
Yeah, I was. I had never lived in a small town, and actually, my dad did grow up in a small town, and he would say, "Oh, small towns are terrible. It's just people gossiping about each other." But I actually quite like it, and it's true. You can't go anywhere without running into people you know.
Yeah. But
there's a nice side to that too. You're never really alone.
Do you fish?
No.
Because
I don't. Being on the water is beautiful, but I don't really look forward to actually catching a fish. So I've never tried it.
I know a lot of people there fish, obviously. Marshall's a fisherman, and then the last time I was there, I went with Dixon Despommier, and he took me on a float. And Neilchi's husband's company, they took us. And I just sat there and watched. I took movies and I didn't fish, but it was kind of interesting, but not something I'd want to do. It's very pretty.
Yeah, I think being on the boat and on the water is just as much fun probably as the fishing. It's a lot more relaxed if you don't have to worry whether you're going to catch something or not.
Yeah, and Dixon would say, okay, you see those moths coming out of the woods, they fall into the river and then the fish eat them. It's kind of an ecology education, really. The ecology of rivers is quite interesting. So I enjoyed it.
In your bio on the RML site, it says you worked a little bit on the 1918 flu that was resurrected from old samples. I wonder if you could very briefly summarize for me, because I remain confused about this. There were a lot of experiments done when that virus was resurrected to try and understand whether or not it was really a more pathogenic virus than other influenza viruses, and if so, why? And, my take-home message from that has either faded or I never understood it. And I'm wondering if you could give me your perspective on that.
Yeah, so it's actually a shame that that this is not used more, I think, because I think a lot of questions still haven't been truly answered. So we know that the virus, yes, it is more pathogenic than if you would compare it to a seasonal flu. It replicates a little bit faster in animal models and in vitro as well, and it causes a little bit more of an inflammatory response. So we know that, but we really don't know, I think, the details of how it would compare exactly, and what exactly are the precise determinants of why it is more pathogenic.
Okay.
And of course, it's really hard to also take everyone is always talking about all of the other circumstances in 1918, of course, and how they would affect this.
Right.
And you see that now too with the coronavirus, right? Older people are more susceptible in this case. But no, I think that the 1918, that's actually one of the reasons why I think it's really interesting. There aren't that many respiratory viruses that cause severe disease if you don't have an underlying condition.
And my recollection also from those studies is that one of the problems was in the studies that tried to look at the pathogenesis, trying to compare results from different animal models. And I'm wondering if you can, what do you think is the best animal model to do that in?
So, well, that really depends. I think for flu, the best model is probably the ferret. So the ferret really recapitulates best the tissue tropism, for example, that you see in humans as well. And that's one of the problems that, of course, in ferrets, we have a lot fewer reagents to look at, or at least at the time when most of this work was done with the 1918, there weren't that many reagents. So for certain aspects that you could study, the non-human primate might be a good model, but we know the receptor distribution is not actually as much that similar to the one in humans. And then, of course, mice also have a receptor distribution problem.
So let's talk about SARS-CoV-2. You published a paper not too long ago in Nature on using Rhesus macaques as a model. And I'm wondering, how soon after you learned about the virus did you start doing those experiments?
So when I guess we heard at the same time as everyone else at the end of December that there was something going on, and at first we weren't sure, and I was a little hesitant because at the beginning it could be anything. Then early January it became clear that it was probably a coronavirus, and of course we had done quite a bit of work on MERS as well. So I decided that we should look into what we would do. So being trained in Rotterdam, I think I have a pretty good. We're trained really well to just jump on these new things when they happen, and to just drop everything else and to figure out what you need to do, and what the interesting questions are, and how you could figure those out. So when we found out it was a coronavirus, we started making a list of things that we would need. Of course, you need the virus, but there's other things that you absolutely need, which is a diagnostic assay, reagents like antibodies so that you could do histology and those type of things. So we had a list made, and that was all divided by, okay, as soon as a sequence is online, we have to do these things. We'll order custom antibodies, we'll order all of this stuff. And then as soon as there is a virus isolate, we'll do this. Because, of course, there also you need to set up all of your assays. So we had that list ready and before the sequence came out, and then we just started going down the list. And then, of course, we were really anxious to get a virus isolate. And now looking back, it didn't take that long, but it felt like it took forever. But we got the isolate on, I think, February 5th.
And where did you get the virus? How did you get the virus?
So we got it straight from CDC.
Okay.
So they sent it to BEI, who then grew it up, and then we were the backup. So we were also growing it up in case they ran into some issues at BEI that we could supply. So when MERS coronavirus, we were the first, because of our link, of course, with Erasmus Medical Center, we were the first in the U.S. to get MERS coronavirus, and we then sent that on to everyone.
I forget what BEI stands for. That's an NIH repository. Is that correct?
Yeah, I also don't know what the letters stand for, actually. But yeah, so you can request viruses, but also other reagents as well. So they now have our non-human primate convalescent serum, for example, that people, if they need a positive control serum, can use for their assays.
So part of what I was thinking when you were talking was that one of the things you would need to do these experiments, and that you were sort of lucky, was that you would need to have the facilities where you were able to work with these types of viruses. Is doing this sort of, rapid change on different viruses something that most people who have BSL-4 capabilities are set up to do, or is that just sort of because you were trained in the Netherlands, in Rotterdam?
I think it's a little bit of both. Yeah, we are definitely, it's definitely one of those things, like Vincent said, all of those different viruses I've worked on is because I really love just dropping everything in, going after this thing that is new and exciting. And I think some people are really more focused in that one molecule or that one virus that they've been studying their whole career. So I think it's a little bit of both. Of course, we are NIH, so we have the ability to spend our money in any way on any type of research, as long as we have scientific productivity. So it's really easy for us to drop everything, because we don't have to ask permission to spend our money on a different virus, like maybe you would have when you are under a grant system where you have applied for money to do very specific experiments.
I'm envious. I'm very envious. So I have to ask you this: in these early days, did Tony Fauci call you up and say, please work on this?
He did not. Good. No, I think we, when this started so we communicated with his office really early on, I think in the second week of January. And then we were just in all of the phone calls, but they know they don't have to ask us if there's a respiratory virus coming along, we'll be on it. Yeah.
So why did you decide to use this particular animal, Rhesus macaques? I know you had lots of previous experience with it, but what were the reasons for SARS-CoV-2? So
it depends if you're asking non-human primates versus other animal models, or are you asking specifically Rhesus macaques?
No, non-human primates versus other.
Okay. So I think there's one is a really practical reasons, which is there's very few facilities that can actually do non-human primate experiments in high containment. And I think we are extremely well set up for that here. So there is a practical issue that that not a lot of people will be able to do it, but there will be a huge demand just from regulatory authorities and a therapeutic point of view. So that's one really practical side, but there's the scientific side too, where we know from other coronaviruses that the non-human primates often are one of the best models, just because of issues with the receptor binding, as in the mice. We know, so we basically went with what we knew from the first SARS, which is you can infect hamsters, but they don't get sick. Ferrets don't really get sick. And then mice need the human ACE2 receptor. So we also were trying to establish protocols so that we could do all of those small animal models, but that was in a different lab here, and so my focus was the Rhesus macaques. And I think one reason why I took that on is that we know. So I had studied remdesivir. To see if it had any effect against MERS coronavirus. And there we used the Rhesus macaque model, because remdesivir doesn't work in in rodents that aren't genetically modified. So we knew that since remdesivir was effective against MERS coronavirus, we had to show whether it would work against this virus as well. And so then we knew we would need a non-human primate model.
So back to the question that you yourself posed, why particularly Rhesus macaques?
Yeah, so that's actually more a coincidence than wisdom.
Yep.
So we actually had Rhesus macaques here on site, and really soon after, when there was a travel ban from China to the U.S., it also became clear that it would become very hard to import monkeys, and the Rhesus are available locally. So. And that's the same with the African green monkeys are really hard to come by as well. So it's really more hoping that they will work because they are available. We don't really have the sophistication, unfortunately, to know beforehand. All we can look at is whether we think the spike will bind to ACE2 of this specific species. But other than that, we have no way to predict which species will be more suitable.
When you started the studies, you probably didn't even know what the receptor was. Is that correct?
No, we did. As soon as the sequence came out on January 10th, the postdocs here were looking at it, and they said it's probably ACE2. And then Michael Letko in Vincent Munster's lab, within 10 or 12 days, had proven that it was binding to ACE2.
That's incredible, just incredible.
Are there more reagents to work with Rhesus macaques than some of the other non-human primate models?
We usually, it really depends. You just have to sort that out for yourself. And since we use the Rhesus macaques quite a bit, we have an array of reagents that we can use.
So you didn't know if they would be susceptible to infection, right, when you started this?
Well, we were pretty sure that we would be able to infect them, since it looked like the spike would bind to the Rhesus ACE2, but whether it would cause disease, we didn't know.
Yeah. So one issue that I think is interesting is that you unlike mice and other small animals, you can't use a lot of Rhesus macaques, right? You have to limit the number of animals, and so how do you do that? Does it depend on the experiment, or do you usually have a certain number that you would use for an experiment, like in this case?
It really depends. So what we have found in the past when we would do 3 animals per group is that for statistics, that's really not ideal because you actually see quite a bit of variation. So usually our standard number for initial experiments like this, we try, if we can, to do it in 4 animals. And so that's what we did. And then it really depends on the variation that you see within the model. You can, it's really hard to do a true power analysis, because if your power analysis tells you to use 12 animals per group, we can't really do that. So, but we do see if there's more variation, like with for example, with MERS coronavirus in the Rhesus macaques, we would use 6 animals, and that's also what we did when we then studied remdesivir.
So this initial paper you did first, I presume, before the remdesivir yeah. So tell us the study design of this experiment.
Yeah, so it's really, of course, you kind of have to think about what you want to see when you start to model these things. So at the time, we actually didn't know that much. So, of course, this was still really early. We started the study on February 14th, I think. So we didn't know that much. At the time, we knew that people were getting respiratory disease, that some of those were getting acute respiratory distress, that they had high viral loads in nasal swabs, and then also some viral RNA in their throat swabs. And that they on either on a CT scan or in X-rays, you would see these pulmonary infiltrates. That's really what we knew back then. All of those things, losing sense of smell, potential heart issues, clotting issues, all of those things all came later. So then you have to, so you want to induce respiratory disease, so you have to decide how you're going to inoculate. So with these new viruses, we then usually try to target the upper and the lower respiratory tract at the same time, as well as mucosal surfaces that could be important for infection in real life. So we usually just do a combined inoculation, intranasal, intratracheal, and then in the mouth and in the eye, just so that you hit all of those at the same time.
And that seems to be a procedure that has some history with you, back to MERS and stuff. Is that correct?
It's even from before that. It's was done for the 1918 flu in primates, and even before that. Yeah, so it's really this is the trouble with non-human primates. When you do these experiments initially, we just usually decide to use all of those routes, just to increase the chance of seeing something. After that, though, it becomes really hard to go back. Right?
I see. Yeah.
Yeah. I can imagine.
And I think deciding whether you want to use more animals and more resources to figure out if you could do more sophisticated inoculation routes is a tough decision.
How do you decide on the dose?
Yeah, so that's also not very sophisticated. We usually just try a high dose. In this case, the virus stock that we grew initially didn't have a super high titer. So we just went with 4 times 10 to the 5 TCID 50 per mil. And we usually, for these viruses, we usually just use a high titer. Again, we can't really afford to try different doses. So you're just hoping by putting in a significant amount of virus that you will actually be seeing disease.
So you did this with 6 animals, I think you said, right? In this.
So 2 groups of 4. So we decided to do 2 groups of 4, and that was so that we could actually do 2 things. One is we could study these animals throughout the infection if they would get sick, but also that would allow us to take animals at an early time point, so that we know that for these respiratory viruses, usually the virus replication peaks really early. And after that, you get, the disease really lags behind that. So if you want to find the virus replicating, you need to sample the animals early. But if you want to look at the disease, then you need to keep the animals in the study. So we had a group of 4 animals that we decided to euthanize on day 3, and then we had another group of 4 that we decided to keep for 21 days. And that was so that we could see if they got disease, we could see them recover, but also if they would not develop disease, then we would have them in the study long enough to see if they actually would seroconvert or not.
And so what did you find in those 2 groups?
So all of the animals get respiratory disease. So they get increased respiration, and you can see that they have to breathe a little bit harder than they normally do.
Had you decided on day 3 when you initiated the study, or did you? Okay, but you couldn't have anticipated what the disease progression was going to be.
No. But this is the day 3. So like I said, we know that the virus replication peaks really early. And so the day 3 was really chosen so that we would be able to find virus replicating if we didn't see much disease.
Okay. And were they showing clinical signs at day 3?
Yes. Already on day 1, you could see a little bit, their respiration going up a little bit. And also they just didn't eat as much, and they weren't as active as they normally are. Then we also took x-rays, and we could see these pulmonary infiltrates like we do in humans. And then we do clinical exams in the first week every other day. And so we could take swabs, and we were analyzing all of those in real time, so we could really see that the virus was replicating. There was very high viral loads in nose and throat swabs. And then we also took bronchial velar lavages, and there was quite a bit of virus in those as well. So then we knew that the virus was probably replicating in the lungs as well.
Did you sample blood? Any evidence of viremia?
We did sample blood, and there was no viremia at any time. We've done more animals since, and it's really we haven't seen it.
Okay.
Which is not, for these respiratory viruses, is not unusual not to see viremia unless the disease gets very severe. And I think that's what you see in humans too, where you will see viremia, but usually if you have a moderate or mild infection, and even if there is lung involvement, there probably just isn't enough damage really to get that leakage into the lung.
Okay.
Or alternatively, it might not be a continuous viremia. And that's something that we don't really know. It's something that we've wondered about with the 1918 flu, because their 1918 flu, in ferrets, they are not viremic, but then you do find the virus in the liver, which makes you wonder how it actually gets there.
It just as an aside, one of the things I found interesting in the data is if you look at the virus load with days after infection, there were occasional, actually not infrequent, but occasional samples where in an animal that obviously, if you look at the whole time course, was carrying virus the whole time, you'd get occasional negative samples. And it reminds me of these situations where people have a negative test and then a positive test. And I was just thinking about the sampling issues.
Yeah. So that's really one of the things that made us really happy about how well the model actually repeats what you see in humans. So this shedding pattern, like you say, especially in the throat swabs, where they will be positive one day and then negative the next, and maybe even 2 days later still negative, and then all of a sudden positive again. It really is something that is seen in humans as well.
And do you think that's just a sampling issue?
I don't think so. I would. I think. Well, it could be, but when you look at the loads that we find, they are actually quite high. So they are still 10 to the 5 copies per mil or something, right? So you would think that going from 2 consecutive negative samples to a positive with such a high load, I mean, to me, that just seems unlikely. If it's sitting around, then you should at least pick up something. So the question, of course, is where that virus is coming from. And that could have something to do with why you're finding it one day but not the next.
You know, what's interesting is that the BALs are pretty uniform. And so there, you know, you're putting fluid in and out, whereas the nasopharyngeal, are those nasopharyngeal swabs? Yeah. So that's, you know, Daniel Griffin always says it's always hard. Maybe you miss the patch of mucosa that's infected one day, you know.
And with the BAL, you did both RNA measurements and you actually did looking at live virus. Is that right?
Yes, and we did that for swabs too. And so what we see is really that the infectious virus is around only for a few days, and after that it becomes really hard. So by day 7, we can still detect some in the lungs if we try really hard, but most of the virus we can detect between day 1 and 4.
And that's just because it's dropping in titer, right? Probably.
Yes, yeah. Yeah, our cell culture is not super sensitive.
So does that tell you something about whether or not, you know, the RNA that you're seeing is always coming from live virus, or do you just think that you're not having a sensitive enough cell culture assay? Yeah, so I think it's a little bit of both. So our cell culture is definitely not as sensitive, I think, as I would like it to be, because even on day 7, when we have quite high genome copies in the lungs, we have a hard time consistently detecting virus throughout the lungs. But we also know that RNA sticks around for a really long time. So it's just really hard, and I think that's unfortunate that, of course, a lot of labs can't do tissue culture, but it would be really interesting to see that comparison in human patients as well.
Yeah.
Do you know why it sticks around for such a long time?
Well, I think part of the problem is that it doesn't have to be. So normally we say RNA isn't very stable, but of course for your QRT PCR, you only need to detect 70 nucleotides. So in a 30 kb genome, you can afford a lot of degradation before you would not detect it in the PCR anymore.
Yeah. So you know, these reports in people where they're PCR positive for weeks and weeks and weeks, you think that's likely just pieces of the genome. There's no replication at that point, right?
I mean, that's likely, but I'm sure there's exceptions.
Yeah, of course.
Because you just don't know what's going on in an individual patient.
Yeah.
So I think unless you have the ability to do tissue culture, it's really hard to decide whether a person is still infectious.
Yeah, sure. So you characterize this as a mild infection, right? None of the monkeys died, correct?
Yes, that's correct. Yeah, they all recover.
So obviously in people, not everyone recovers. So were these young primates, relatively speaking?
Yeah, so they're 3 to 5 years old, so that they're really healthy young adults. And I think so. We have actually tried to see if we could make a more severe model using different non-human primates, and we just haven't been able to yet. So we are now going to try older Rhesus, which the problem with that is they're not so easy to come by, right? Even if you would see severe disease, it wouldn't necessarily be a solution for the lack of a severe disease model.
Yeah,
which of course we really need a severe disease model, because that's what you need to test all of those host targeted therapies. When we put our preprint online on the Rhesus model, I got a ton of emails. Can you test anti-IL-6 for me? Can you test this? Can you test that? But none of those host factors really play a role in the non-severe disease that we see. So we really need a model. So we are going to test older Rhesus, but we're going to do it more from the point of view of trying to understand if we see the severe disease. We really want to try and understand why we see more severe disease, so that maybe that could guide us, in trying to model it differently, but also in trying to figure out where the therapy should be targeted potentially.
There's no model at the moment for severe disease caused by this virus, right?
Yeah, I think the mouse model is coming down the line.
Yeah.
And so that probably, of course, there's a good chance that will be more severe.
Yeah. Yeah. Interesting. Do you think, now, considering your history with transmission, would this macaque model be a good transmission model, do you think?
Yeah, we've talked about that. And unfortunately, we can't really do those studies right now just because we're prioritizing more the vaccines and antivirals.
Yeah.
But yeah, based on the amount of shedding that we see from the upper respiratory tract, I'm pretty sure it would transmit.
Yeah.
You may have already said this and I missed it, but what sort of containment is used for these sorts of experiments?
So you can do these at BSL-3, but we don't have the ability to do non-human primate experiments in BSL-3 at RML, so we do them in BSL-4.
Okay.
Wow. So you have to get all suited up. Huh?
Yep.
But I kind of prefer it. I kind of like the shower at the end.
Well, you're not the only. We've talked to several people who work in BSL-4 facilities who've told us that they feel safer there than at lower levels of containment, just in general. Yeah,
I think that for me, that's not really. I don't feel necessarily safer in the 4 than in the 3. It's just the way our lab is set up in the 4. It's just really set up to do these type of studies, and our BSL-3 is not. And so I think it's just, and we have a lot more space in the 4 than in the 3, so it's just a lot easier to just do. We have more equipment, which is really well set up in the 4. And we are all used to going into the 4. So whereas other people are, oh my God, it takes 20 minutes to come out. For us, it's part of life.
Yeah.
So
get used to it. So you now have a preprint on bioRxiv of the remdesivir. Now, did you start that at the same time, or did you wait for the first experiments in your macaques?
So we started as soon as we knew that there was disease in the Rhesus and that we had good levels of virus application in the lungs based on the bowel. As soon as we had that, I put in an animal protocol for the remdesivir study, and then we started. I think about 3 weeks after we started the Rhesus, we did that study. Yeah.
And you give the monkey, in that case, you give them drug 12 hours post-infection, then daily afterwards, right?
Yeah, so they that's based on the fact that when you look at the bowel, really the peak of replication is probably maybe day 1, maybe day 2.
Mm-hmm.
So really early. And since remdesivir inhibits replication, you want to hit the virus before the replication has peaked, or it'll be useless.
Yeah.
So we decided to treat at 12 hours, and then again at 24, and then every 24 hours after that.
So they're getting. Do you know if that compares to the dosing that's used in humans? I realize that in humans it's you can't start it 12 hours after infection, but in terms of the dosing protocol, it's the same.
So the dosing is comparable, and so in humans, of course, in non-human primates, we dose them per kilogram body weight, which you don't do in humans. So humans get 200 milligrams loading dose, and then 100 milligrams per day after, and we give 10 milligrams per kilogram loading dose, and then 5 mg per kg after. And that's really based on Gilead has done the comparison of the PK data, so that it looks like they would be dosed similarly.
And I was curious. You talked about looking in the animals for the metabolism of the drug, and it was the first time that I'd really looked carefully at the structure, and I noticed that the drug has it's a nucleoside analog with a funny ribose and a funny base, and then a 5' phosphate and a whole bunch of spinach hanging off of that 5' phosphate. And it looks like it actually gets metabolized to a triphosphate, which would be the active drug in vivo. And I was wondering if is that correct? And second, do you know which components of the drug are the active components? Do you know how it works?
You should especially address the spinach.
Yes, that's probably the thing I'm most knowledgeable about of everything you just said. So unfortunately, I haven't done any chemistry in this century, so I don't really have the metabolism. So we worked with someone to really look at the PK, and he figured out all the metabolites. And all I hear is phosphate, when he talks about it. So I can get you the information. I just don't.
Okay,
ready.
Fine.
So this was relatively effective at suppressing disease and viral loads, but not in the upper tract, right?
Yes, that's correct. So what we see is really that we don't see that many pulmonary infiltrates. The animals, they don't really get the obvious disease signs. The loads in the lungs are lower, but when you compare the swabs, they are identical between the two groups. So the question then is why? And we don't really know. I think the most likely option is probably that the drug is not getting there efficiently enough. We know that the concentration in the nose of the drug is lower than in the lungs, but we haven't been able to test that on our animals.
Got it. And you just did the one. You just started at 12. You don't have any idea how long after infection you could give drug and still have an effect, right?
No, and the reason why we didn't do that is because we knew when we started, the first clinical trials were starting too, and because it's an intravenous drug, we knew that would be they would be treating patients that were a little bit further along in the disease process. Yeah. So we thought if we include those studies, we probably get bypassed by the clinical trial data that come out. And so it seemed a little bit of a waste.
Yeah, that's a good point. Because in humans, I think they're treating much later than 12 hours, probably days, right?
Yeah, and you can't really compare that either, right? Because we give a super high dose, the animals are sick. There's not really an incubation period that way either. And then the disease peaks a couple of days later. So how that compares to a person who has to be hospitalized before they would be treated is really hard to say.
Sure.
Nevertheless, you saw a pretty pronounced, obvious therapeutic effect, whereas my understanding is that the effectiveness in humans is not nearly as obvious. Would you attribute that to the timing?
Yes, definitely. So I think by the time they treat humans, they might be already at the peak of infection or past that. and of course, and we are well before, so we still can reduce, and that's what you see in our data too. When you look at the amount of virus in the bronchoalveolar lavages, even 12 hours after the first treatment, the virus titer in the bowel is 100-fold lower.
Yeah.
So if you hit that on time, then obviously you can really reduce virus replication. How much effect you can really have later on, obviously the human data show that it's probably quite limited.
Yeah. So that's really very informative, okay, knowing that this drug actually works in a primate.
Yes.
Okay, and that the timing is a critical issue.
Yes, and that's exactly why we did the study. So I was really worried because, of course, it just so happened that I had just written up the MERS remdesivir manuscript in December. So in January, when we found out it's a coronavirus, I started sending the manuscript to the WHO and the U.S. government, just making sure that people were really aware that this was a potential drug. But what I was really worried about, because this is what happened when we when MERS emerged, we developed an animal model, and then Daryl Falzerano in Heinz Lab showed that if you treat them with interferon and ribavirin, then it has a clinical effect. Then they decided to test that in humans that were really, really in the very last stage of disease. And then concluded that it didn't have any effect. And since then, I think that's kind of been ignored as a treatment option. And so I was really worried that would happen with remdesivir too, that we would say, we think it might work. And then they would wait till the most severe cases, they would try this treatment option. And that's why I think it was really important to show that if you treat early, then it actually has a very clear effect.
Well, it's, and it becomes, I think clinically, it becomes a real problem, in particular, if the supply of the drug is limited, is trying to, the triage. Who do you treat? Who do you not treat? Daniel spoke about this on his episode that dropped on Sunday.
That's right.
I agree. And I think that's a real issue. And I think ideally there would be plenty of drug, and ideally you could maybe administer it via a different route, where you could administer it, really soon after positive diagnosis and actually prevent people from being hospitalized. At least in our Rhesus macaques, you can really, when you look at the lungs histologically, the treated animals, they don't develop that interstitial pneumonia that we see in the controls. So if you could treat really early, of course, there are some issues with treating with these type of drugs that might have side effects and all of that. But if you could treat early and prevent hospitalization, that would be ideal, I think.
So you also do the Rhesus experiments with intravenous drug. Is that correct?
Yes. Yep.
And have people done experiments with the drug delivered with any other way in the macaque or any other model?
So we are going to try that, yes. But the problem is because of there's only so many. So apparently, oral administration doesn't work. So I don't know the details of that. I just know that it's not an option. So there's very few options that you can try, and then you probably there could be a volume issue or a vehicle issue. It's not, the remdesivir is not super soluble. So that's something that Gilead is figuring out, actually trying to see if there's a different formulation.
I would think that would be high priority, because if you could take this as a pill at home, it would make a huge difference. And it's not like you can't do that with nucleoside analogs. After all, that's the way the HIV drugs are done.
So the way I understand it with remdesivir, it doesn't work orally, but I don't know the details. There's other options.
There's NHC is another one, right? Which is similar, and that is bioavailable, right? You've probably heard of that one.
Yes. Yeah.
So it's possible.
That's just not, they are just now going into safety testing in humans with that. So that's a little bit behind.
You haven't looked at that in NHPs, have you?
No, we haven't. No.
Okay.
Others are doing that, actually.
So, I mean, you got this bioarchive on SARS-CoV-2. You just published the MERS and remdesivir. Last year, you published a Nipah remdesivir. So what is it with remdesivir?
So the funny thing is, it all started with MERS.
Uh-huh.
So with MERS, we were really one of the few people, I think, because that didn't explode the way, of course, SARS-2 now did. There was interest in the coronavirus. People were interested, but not a lot of other people. And there's just very few people who can do these type of experiments. So we were asked to test a couple of antivirals. So we actually did some antibody testing as well. And then we tested remdesivir. And when I was talking to Gilead, they were saying, we think this remdesivir actually crosses the blood-brain barrier. Mm-hmm. So then I immediately thought, this would be perfect if that's true, and it would work for Nipah. That would be perfect. So I told them, I'm really interested in Nipah, scientifically more interested in Nipah than in MERS coronavirus. So should we not test it for Nipah? And then they said, well, we're working with other people on that. But that kind of fell through. I think Nipah is one of those, not a lot of people get it, mm-hmm. but if you get it, doesn't look good for you. So I think I was just really interested in pursuing it for Nipah as well. And that's what we did. Yeah. And then, when SARS-2 came along, and once it started spreading on such a big scale, it's such an urgent question. And this, based on what we knew for MERS coronavirus, this was one of the most promising, mm-hmm. I would say, therapeutics. So
now the Nipah study was done in African greens.
Yep.
And is that because that's the Nipah model?
Yeah. So for Nipah virus, they actually they are the non-human primate model that really recapitulates disease best.
Got it.
So with Nipah virus, the animals actually really get acute respiratory distress, mm-hmm. and it's actually really acute. And so, yeah, that's the best model, since you can't test this in small animal models. Mm-hmm. There's a transgenic mice where they knocked out part of the drug metabolism so that you could use remdesivir, but then mice aren't really susceptible to Nipah virus.
Right. So what's the status of remdesivir for human Nipah infections?
So the problem there is, after the outbreak in Kerala in India, mm-hmm. NIAID started working to prepare these clinical trial protocols. Mm-hmm. And number one right now is the monoclonal from Chris Broder's lab, the M102.4.
Right.
They have done, they have phase 1 data, and it really looks like the most promising treatment, even compared to our remdesivir data.
Got it.
So that's at number one. That now, of course, that remdesivir is going into humans on a larger scale, I think that really increases the chance of that being added. Mm-hmm. But what was discussed quite lengthy in Singapore was that we should probably test a combination of the two in the African green model. Mm. But obviously we haven't gotten around to that yet because we got busy doing other things.
Yes, quite clear. I wanted to do one more thing, and I know we've been an hour already, but let me ask you. Then there's there's a bioarchive of the ChAdOx trial.
Yes.
And what's your role on that one? Did you do the animal work?
I helped a little bit with the animal work, and other than that, I'm just more of a. So I helped a little bit with some of the work inside, and other than that, mm-hmm. I just think along, I guess.
So this is the same model, right? It's the model
yes.
you started with, except here they're immunized beforehand, correct? With this chimpanzee adenovirus spike construct, correct?
Yeah, so they're immunized 4 weeks before challenge. And I have to say, one thing that people might overlook is that, of course, we're quite limited with the animals sometimes that we get into the facility, especially now that everything has to be done in a hurry. Mm. So when we did, when these animals were vaccinated, the only Rhesus macaques that were available were really young Rhesus macaques. Mm-hmm. And I think that's part of why the data look, in some of the aspects, a little bit different from what we saw, because our animals were bigger and older. Uh-huh. Yes, but yes, they were vaccinated and then challenged 4 weeks later.
And challenged by the same multiple routes that you did before, right?
Yep. So that was all done the same.
Okay.
And how do you deliver the vaccine?
It's just an IM injection.
And this reasonably protected against pulmonary pathology, right?
Yes. So I think there again, the question is of course, what are you trying to achieve with a vaccine? And we would all love for a vaccine that provides sterilizing immunity, but that's a pretty high bar to set.
Yeah.
And these animals, they do show less clinical signs of disease, and they also have lower viral loads in the lungs, and then we also see less pulmonary damage.
Yeah, I would be surprised if any vaccine against this virus is sterilizing. Given the history of other coronaviruses, it'd be good if they just prevented death.
Yeah, so the question is, of course, if you could maybe administer the vaccine intranasally, and would that be better?
Yeah.
There's still, this is just the very first experiment, and in all fairness, one of the main reasons why this experiment was done was because they needed these data to start clinical trials in humans. So
right.
One of the big issues with vaccines is the enhanced disease.
Yeah.
If you've been vaccinated and then you become infected, that you would get more sick than if you hadn't been vaccinated.
Mm-hmm.
And that had to be shown that was not the case, which was one of the main reasons to do the study. And of course, it's really nice to see that the animals also, not only did they not get more enhanced disease, but also they didn't actually get a pneumonia.
Right. So
relevant, I think, to that point, and I don't know if you can speak to this, and maybe Brianne can weigh in on this. There's this sentence in the paper that says that, this is from mice, but it says in the mouse experiments that the immune response was not Th2 dominated, demonstrated by IgG subclass and cytokine expression. And if my understanding is that this Th1, Th2 response is relevant to whether or not there might be enhanced disease, but I'm vague on it. Can you speak to that at all?
Only a little bit. So maybe Brianne should weigh in. So I think that this has been shown in the past that with a predominant Th2 response, for example, when they started vaccinating kids for RSV and measles in the 60s, some of these kids then got infected and they got more severe disease than kids that were not vaccinated. And so in those cases, it's been shown that a predominant Th2 response is what caused that.
Yeah, that's basically pretty much what I would have said as well. The Th2 cytokines, the Th2 cells that are activated may seem to lead to a little bit more damage than the Th1 responses. And so I don't know if I would exactly call it enhancement. That's what you can argue about what you call enhancement, but the Th2 responses do seem to lead to a little bit worse disease. And so trying to determine whether you are seeing more of a Th1 bias response could be really important. And that seems to be really different in different mouse models. And so that's also key here.
And so in my sort of immunologically naive perspective on this, Th2, I'm looking for education here. Th2 means to me a response that's skewed towards the antibody as opposed to the cellular response. Is that fair, or is that an oversimplification?
That's sort of an oversimplification, only because, and the reason why is because we now know about more than just Th1 and Th2.
Oh God, don't tell me.
Yeah. So when I think of antibodies, I think of one of the others. The Th2 response has a lot of IL-4 and IL-5, and it is the one that we see in things like asthma and also in respiratory allergies. And there are things like neutrophils and mast cells that are mast cells in particular that are playing a big role.
Nevertheless, it does speak to the point that you have to be careful about what your vaccine actually does, which is why they're looking at this, sort of what the overall profile of the immune response is, in particular if you're concerned about something like enhanced disease.
Yeah, I always think I always, say to students that it's not about having a vaccine that makes any old immune response. You actually have to have a protective immune response.
Right.
So this, Emmie, was important in having the phase 1 proceed with this vaccine, right?
Yes. And they're now actually just, I heard today, they're moving forward with the phase 2 now.
Cool. Very good.
Yeah.
I'm curious, did they approach you or did you say, hey, we can do this?
So this is all being done in Vincent Munster's lab, who's also here at RML, with Neeltje, who you mentioned earlier.
Right.
And they already had a CEPI project for MERS coronavirus and Nipah vaccines based on the ChAdOx vector. So there, again, this was like the remdesivir story, right? You're working on this vaccine, you hear about this new virus, the first thing you do is email ChAdOx or the ChAdOx, the people that make the vaccine, the Oxford at Jenner Institute, email them and say, hey, did you hear about this new virus? Should we try a vaccine?
Anything else, Rich, Brianne, that you want to
I could go on forever. This is great. But nothing pressing.
All right.
No, I'm learning lots of things, but I can learn another day, I suppose.
All right. That's episode 622, microbe.tv/TWiV. Questions and comments, TWiV@microbe.tv. If you like what we do, consider supporting us, microbe.tv/contribute. Our guest today from the NIH in Hamilton, Montana, Rocky Mountain Laboratories, Emmie de Wit. Thanks so much for joining us today.
You're very welcome.
And thank you for doing this work. It's really critical and important and wonderful, and we do appreciate it. So thanks for that.
Thank you. Thanks.
Rich Condit, Emeritus Professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Thank you. I learned a tremendous amount here, and I second everything you said. It's important work. Thank you, Emmie, for the work and for being with us. I really appreciate it.
Thank you so much.
Brianne Barker is at Drew University, on Twitter, BioProf Barker. Thanks, Brianne.
Thanks. It was great to be here. And thank you so much, Emmie. This was really fascinating, and it's a great thing that we have people doing all this work. So thank you for all of this.
Thank you.
And I'm sure there's a lot of stuff going on, and in 6 months or so, maybe we'll have you back after you've published 10 more papers.
Yeah, that'd be great.
Well, I'll just set the bar really high, but I'd be happy to explain more, yes.
I'm Vincent Racaniello. You can find me at virology.blog. Like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.